Okay, biologists, let's review transcription and translation. So the whole point of transcription and translation is to use it to illustrate what we learned about biology called the central dogma, and that is that on your DNA you have genes, and those genes contain the instructions to make proteins, and those proteins are what give you your traits. Those traits could be as simple as your eye color or your hair color, or whether you are lactose intolerant or not. There's a lot of characteristics about you that make you you. These aren't always one-to-one things. It's not like there's one gene that determines your height or one gene that determines your hair color. There are some pretty simple ones, maybe lactose intolerance, because it's just about one molecule. Maybe that one is a little bit more simple. But regardless, this is what we're going to be doing. So we are going to be talking about how genes can be used to produce proteins inside of your cells. This happens in a two-step process. The first step is transcription. Step 1. This is transcription. Transcription occurs in the nucleus of the cell, and we can see it in our picture here. This is the nucleus. Here is our double-stranded DNA molecule, and you can see that this DNA molecule gets pulled apart. It gets pulled apart. So that is step 1. Step 1 is that part of a DNA molecule unwinds and the weak hydrogen bonds between the complementary base pairs break. This exposes the gene to be transcribed. So when this process happens, it's only the gene that your cell is trying to produce the protein of that is transcribed. If you have questions about what a hydrogen bond or a complementary base is, I would go back to our DNA drawing video. Okay, step 2. A complementary copy of the code from the gene is made by building a single-stranded nucleic acid molecule known as mRNA. So mRNA is this molecule here, this single strand that is being made as a copy of one of these DNA strands. All right, sorry about that. You don't know that, but I had to pause that. My house is loud. Okay, mRNA. So a couple things to remember. In DNA, the base A always pairs with the base T, and the base G always pairs with the base C. This is called complementary base pairing. However, RNA does not contain the base T. So, if the DNA strand has an A on it, the RNA strand will have a U on it instead. If DNA has a T, RNA can have A, that's no problem. If DNA has a G, C, and C, G. It's kind of common for students to say that A turns into T or something like that, but that is not proper use of our verbiage here. You should be saying that the complementary base for A is U on RNA. All right, let's go to step 3. There are little floating RNA nucleotides in the nucleus, and they pair up to their complementary bases on the template strand of the unzipped DNA molecule. Here is our little free DNA nucleotides just floating around in the nucleus. And so when a blue DNA piece is read, a green RNA nucleotide is put on this RNA. And this just keeps going on down. When the gene has been completely transcribed, and that is, transcription is the process of making this mRNA molecule, the weak hydrogen bonds between the mRNA and DNA strands break, the mRNA separates from the DNA, and the double-stranded DNA molecule reforms. The last bit is that the mRNA molecule leaves the nucleus through a pore in a membrane, and this is where the term messenger comes from. The mRNA is sent, carrying a message, to another part of the cell. DNA can't make this journey. It's too big to fit through the pores in the DNA membrane, or in the nucleus membrane. So the whole point behind transcription is that a part of DNA is read, and that part is what we call a gene, and we end up with a mRNA copy of that gene. And this mRNA is going to leave the nucleus, and the next part happens. What is that next part? That is step 2, and this is called translation. Step 2. So, in this central dogma of genes to proteins to traits, we are halfway through. We have done transcription. We are now going to do translation, and that is where we have our protein being produced. All right, let's do the same thing with this here. Let's read a description. Let's take a look at our picture model here, and then we'll do one for an example. So, transcription is, there's a typo I just caught. Oh my gosh. This should say translation occurs in the cytoplasm of the cell. Remember that our mRNA strand here is going to leave the nucleus and come out to another part of the cell. That part is called the ribosome. All school year we've been saying that the ribosome is where proteins are produced. That's what we're going to talk about now. All right, so step 1. After transcription, the mRNA moves out of the nucleus into the cytoplasm toward the ribosome. Step 2. After leaving the nucleus, the mRNA attaches to a ribosome. Ribosomes themselves are made of proteins and RNA. Step 3. Translation depends on complementary base pairing between codons on the mRNA and anticodons on the tRNA. These two words are really important. Codon and anticodon. A codon is 3 bases on the mRNA. The anticodon is the complement to the codon. Complement means that those pairing rules apply. If the codon has a G, then the anticodon will have a C, and vice versa, and so on and so forth. Though, in the cytoplasm, there are floating molecules of something called tRNA. And those tRNA molecules are attached to specific amino acids. Take a pause really quick. Remember that amino acids are what make up proteins. Specifically, when we chain amino acids together, we end up with what's called a protein. So, each triplet of bases pairs with a complementary codon. Each triplet of bases on the anticodon pairs with a complementary codon on the mRNA. And those codons are 3 bases. The transfer RNA brings the amino acid over to the ribosome. The amino acid can then be bonded onto this polypeptide chain. A polypeptide chain is a chain of amino acids. Amino acid, 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 amino acid. This whole chain is what we call a polypeptide chain. Certain codons carry the command to stop translation when the polypeptide chain is complete. These are called stop codons. And when a stop codon is reached, the polypeptide chain is released. It will be folded into a very specific shape, and it is now a protein ready to do work. Uh-oh. No, I'm not trying to draw. All right. What is going on here? All right, here we go. So, we've got our mRNA strand. It comes out here to the ribosome. On the ribosome, the mRNA, this is the mRNA molecule here, mRNA. It gets kind of pulled through the ribosome, kind of like if you're pulling a string through a hole. Like imagine your shoelace and you're pulling it through a hole. The hole would be the ribosome and the string is the mRNA. Every 3 bases, And this one is UGU here. Every 3 bases is called a codon, and that corresponds to an anticodon. It's not labeled right over here, but I'll write it down here. This is an anticodon on a tRNA molecule. So U is complementary to A, G is complementary to C, and U is complementary to A. This tRNA brings with it an amino acid. That's tyrosine is one example of an amino acid. And then the next codon is read, ACC. The tRNA that comes over will have a UGG on it, and that brings with it another amino acid. And so on and so forth until we get this big chain of amino acids forming. This is our polypeptide chain. There's another amino acid and another amino acid, until eventually one of these codons says to stop. And this polypeptide chain right here ends up becoming a protein. And my dog hears my voice and is coming back down to say hello. Hello, dog. All right, let's pick this up. How does this whole process work? Well, let's do it. I got a little model here. This is a model for how we could show protein synthesis right here. Let's start off by making our DNA strand. So DNA, remember, has 2 strands to it. This first strand is what we're going to call the coding strand. Coding strand. So let's start way down here. And if the DNA base on the coding strand is an A, the complementary strand will have a T. And an A, and a C, and a G. I want you to pause the video and fill this in your binder. And come right back as soon as you're ready. All right, I'm just going to go ahead and fill this in. All right, so yours should look like mine. Okay, so let's do transcription now. So we're going to transcribe an mRNA strand from the bottom DNA strand that we just made. This bottom DNA strand is what we call the template strand. A template is something you use to make something else. All right? You may have a template of a project, and you make your project kind of look like that one. That's why it's called a template strand. So, the only rule we have to remember is that there are no T's on mRNA strands. Instead, we're going to use the letter U whenever we need a complementary T. So, if the DNA base is a T, the complement to that for RNA is an A. The DNA base is an A, the complementary RNA base is a U. DNA base is a C, the complementary mRNA base is a G. DNA base is a C, the complementary mRNA base. Whoops, I said that backwards. If the DNA base is a G, the complementary mRNA base is a C. All right, go ahead and do this. You can pause the video and come back to this. Practice transcription in your notes. Okay. This is the mRNA strand. So what we just did was transcription. That is this part up here. We just unwound as a DNA molecule, and we used the template strand to make an mRNA molecule. Now we're going to do translation. So in translation, this mRNA strand is going to go out into the cytoplasm, where a ribosome is going to pick it up, and that ribosome is going to read it 3 bases at a time. Now we call a set of 3 bases a codon. So let's circle each codon just so that we know what it is we're doing here. All right. And what we're going to do is we are going to use a little kind of decoder chart to figure out which amino acids are going to be brought over by the tRNAs that read the codons. So we've got this nifty little diagram down here. And let's do our first codon. Our first codon is AUG. So what we're going to do is on this nifty diagram down here, we are going to start with A as the first base. That's A right here. So now we are only going to care about what is in this quarter of this particular diagram. Our second base is a U. That's our second letter right here. Our third base is a G. That's our third base right there. And this means that the amino acid that the tRNA is going to bring over is called MET. So we're going to write MET in this first box here. So when the ribosome reads this codon, it's looking for a MET amino acid. Now, MET is short for methionine, but we don't need to know that. Our second codon is CAU. Whoops. All right, so I'm going to erase what I just did on this model down here, and let's go back. CAU. That is CAU. Our second amino acid that is put on our polypeptide chain is called his, or histidine. I'll do one more here. UCC is serine, is our third amino acid. So why don't you pause the video and try filling in these other ones. I'm going to go ahead and fill them in while you are working. Lysine is the next one, followed by glue or glutamic acid. Arginine is next. Cysteine. Tyrosine. And then our last one doesn't have an amino acid that goes with it. But instead, there's a little square that shows up. That is our stop codon. Stop is not an amino acid, but it's just a clue for your ribosome to say, hey, we're done making our polypeptide chain. Let's let this polypeptide chain go. All right. And that is the gist of protein synthesis. In our next video, we will go over some of the other pages, exploring how changes to the DNA can cause changes to the proteins. That is called mutation.